Hi everyone, this is the second lecture of topic 5. We are continuing with the skeletal system. In this lecture, we are going to start with the microscopic anatomy of compact bone. The compact bone is made up of units known as osteons. An osteon is also called a Haversian system. So the osteon consists of an opening in the center or a canal-like structure which is known as the central or the Haversian canal. And the central canal contains arteries, veins, and nerves. Perpendicular to the central canal is another canal, that means it is at 90 degrees, and that canal is known as the perforating or Volkmann's canal. It also carries blood vessels and nerves. This figure shows a section of a compact bone with the osteons. You can see the central canal and the perforating or the Volkmann's canal, which are at 90 degrees to each other. In the osteon, around the central canal are concentric lamellae. Concentric lamellae means rings of the bone cells around the central canal. Mature bone cells, which are known as osteocytes, are present in pocket-like structures called lacunae, and the lacunae are arranged in the lamellae. Once again, the lamellae are concentric rings around the central canal. Concentric means the rings, there are multiple rings with the common central point, and these are gradually increasing in diameter. In addition, we also see canaliculi, which are tiny canals or cytoplasmic extensions that connect one osteocyte to another. They also connect the central canal to the lacunae of the osteocyte, and these serve as communication and transport system. This figure shows osteocytes in lacunae arranged in the concentric lamellae. You can also see the canaliculus. Next is the microscopic anatomy of spongy bone. If you remember, spongy bones are lighter and they are made up of network of plates, and these branching plates are known as trabeculae. So trabeculae, they have no blood vessels, but there is a lot of empty spaces between them, and those empty spaces are filled with bone marrow. Trabeculae have lamellae, that means concentric rings, but no osteons, and the function is to create the lightweight nature of the bone. So it's kind of compensating for the heavy weight of the compact bone. This figure shows the lamellae inside the spongy bone. If you are asked to differentiate between compact bone and spongy bone, you will start with the differences in the structure. You know that compact bones have osteons, spongy bones do not. Compact bones are densely packed, spongy bones are not densely packed. But then you'll also have to write about the functional differences. So compact and spongy bones are different from each other both anatomically and physiologically. Functionally, because of the presence of osteon, the compact bone is able to conduct stress from one end of the long bone to the other. But in the spongy bone, there are no osteons, and the trabeculae help the spongy bones to deal with stress from the sides. Now an overview on cartilages. Next to bone, cartilage is the firmest structure. The cells of cartilages are known as chondrocytes. The chondrocytes, just like the bone cells, osteocytes, are located in pockets called lacunae. The ECM of cartilages is semi-solid or gel-like, and it is made up of a chemical known as chondroitin sulfate. The extracellular protein fibers consist of collagen and elastic fibers. Cartilages, unlike bones, are avascular and they have no nerve supply. That means the repair process or healing is very slow in cartilages. Now, if you remember from the lecture on histology, there are three types of cartilages in the human body: hyaline, fibrocartilage, and elastic cartilage. Cartilages have an outer covering which is known as the perichondrium. This perichondrium has two layers. The outer layer is fibrous, it's made up of fibroblasts, while the inner layer is cellular, it's made up of chondrocytes. And because cartilages are avascular, nutrients have to diffuse through the ECM. Cartilages show two types of growth: appositional and interstitial. Appositional means growth from the outside. That means matrix and chondrocytes are added to the outside of the tissue, while interstitial means growth from the inside. So matrix is made in between the cells. Comparing the three types of cartilages, especially their location, hyaline cartilage is present in the connection between the ribs and the sternum. It is also present in the tracheal rings. The hyaline cartilage is made up of very fine collagen fibers. The elastic cartilage is located in the external ears or in the epiglottis, and it contains collagen as well as elastic fibers. The third type of cartilage, fibrocartilage, is made up of collagen fiber, but this particular collagen fibers, they are thick. An example of fibrocartilage, they are found in knee pads. So how does the development of bone occur? In the human embryonic stage, before 6 weeks of development, all of the skeleton is made up of hyaline cartilage. It gradually gets replaced by bones. So we have to learn about three important terms here. The first one is osteogenesis. Osteo is bone, genesis is birth. So this is any process or all of the process that involves bone formation is osteogenesis. The second term is ossification. Ossification means when some other kind of tissue gets replaced by bone. If bone is replacing mesenchyme or fibrous connective tissue, then it's known as intramembranous ossification. While if bone replaces cartilage, it's known as endochondral ossification. The third term is calcification. Calcification means deposition of calcium. So calcium salts such as calcium phosphate, calcium carbonate, they get deposited in the ECM of the bone, and that is known as calcification. So both ossification and calcification are included under osteogenesis. In intramembranous ossification, the osteoprogenitor cells, which are bone stem cells, they differentiate to form osteoblasts. Now remember that osteoblasts secrete osteoid, which is the organic component of the matrix. As the osteoid is secreted, the osteoblasts get trapped in the matrix, and then they mature into the osteocytes. So bone basically expands as a series of spicules, which are projections that spread into the surrounding tissues. The spicules soon start interconnecting with each other, and as they do so, they trap blood vessels within the bone. And initially, the spongy bone is created, but soon areas of spongy bone are removed, and that creates the medullary cavities. And later on, through bone remodeling, the spongy bone can be converted into compact bone. In endochondral ossification, cartilage changes into bone. So what happens is that cartilage cells or chondrocytes, which are present in the future diaphysis region, they start increasing in size, and soon there is calcium deposition in the surrounding matrix. We call it matrix calcification. Now blood vessels slowly start invading in this cartilage region, because remember, cartilage to begin with did not have any blood vessels. Then the cells in the periphery, the perichondral cells, they start forming the outer layer of bone called the periosteum. This is the beginning of a thin layer of compact bone. Now the chondrocytes die because now that the matrix has become solid and calcified, nutrients cannot diffuse to the chondrocytes, and they leave spaces. In these spaces, osteoblasts are formed from the bone stem cells. Osteoblasts would begin to form spongy bone initially. This is known as the primary ossification center. Soon, the cartilage which is present in the metaphysis region, that means the junction between the diaphysis and the epiphysis, it gets invaded by osteoblasts, and the bone starts increasing in length as well as diameter. This figure illustrates what we have just described in the previous slide. Soon, blood vessels and osteoblasts, they migrate to the centers of the bone ends, that means the epiphysis, and there they create the secondary ossification centers. Spongy bone formation occurs in the epiphysis, and now the epiphysis and the diaphysis are separated by a narrow epiphyseal plate. In the growing years, the epiphyseal plate keeps enlarging by interstitial growth, and osteoblasts invade the epiphyseal plate and keep replacing the cartilage with bone. and this leads to an increase in the length of the bone. At maturity, the rate at which the epiphyseal cartilage was enlarging slows down. Also, the rate at which osteoblast was creating the matrix, this also slows down. And ultimately, it comes to an end. So the epiphyseal plate narrows, and then finally it just disappears, and now it is replaced by the epiphyseal line, and this event is known as epiphyseal closure. So epiphyseal plates help a person to grow in height because it allows the lengthwise growth of the long bones during childhood. Bones are very dynamic tissues and they are remodeled all through the growing years. The bones get remodeled basically in response to two factors. The first is the level of calcium in blood, and second is the gravitational pull on the skeleton along with the attachment of the muscles. Bones also grow in width, and that is known as appositional growth. This figure illustrates bone growth and remodeling. So bone growth occurs as the bone grows in length because cartilage is constantly increasing in the metaphysis region as well as at the ends of the epiphysis, and cartilage is also constantly being replaced by bone, while bone remodeling occurs when the growing shaft is restructured, and this is due to bone resorption and also bone appositional growth. All of you are familiar with breaks in a bone, which are known as fractures. Primarily, there are two types of fractures. The closed or simple fractures where the bone break has not penetrated the skin, while the open or compound fracture where the broken bone has come out through the skin. Now, usually the fractures are treated simply by reduction and immobilization. That means giving it adequate rest because bones are highly vascularized, so they heal pretty fast. There are 4 steps of repair of bone fractures. The first one is called formation of hematoma. Hematoma is when internal bleeding occurs. It's a blood-filled swelling. Second is a rough patchwork is done by fibrocartilage. Fibrocartilage formation occurs, it forms a callus. Third, the fibrocartilage callus is replaced by a bony callus. So this is endochondral ossification. And finally, the bony callus, which is still kind of a patchwork, is remodeled. That means bone resorption occurs here to form a permanent patch. Nutrition plays a very important role in affecting the growth of bones. Bones need calcium. So if there is lack of calcium or protein, then the bone growth would not be adequate and bones would be small. Also, vitamin D is required for effectively absorbing the calcium from your food in the intestine. If there is deficiency of vitamin D in childhood, it can cause a disease called rickets. The deficiency of vitamin D during adulthood leads to osteomalacia. Vitamin C is also very important for proper bone growth because this is required for the synthesis of collagen fibers. Right? A deficiency of vitamin C causes a disease called scurvy. Bones play a very important role in maintaining calcium homeostasis. What is homeostasis? Homeostasis is a property of the body to maintain a stable equilibrium. That means to maintain steady internal state. Now you know that the matrix of bone contains deposits of calcium, calcium phosphate and calcium carbonate. So the level of calcium in blood is maintained by the moving in and out of calcium from the bone. Whenever the level of calcium drops in the blood, the osteoclasts become very active and they start breaking the bone matrix and they release calcium. So calcium comes back into blood and it elevates the level of calcium back to normal. And the opposite happens when the level of calcium is higher than required in the blood. Then the osteoblasts become very active. They make use of this extra calcium and they make new bone. So there are 2 hormones that control these 2 actions. These are known as the parathyroid hormone and the calcitonin hormone. The figure and the text on this slide shows the hormonal regulation of calcium homeostasis. Parathyroid hormone is secreted by the parathyroid gland, while calcitonin is secreted by the thyroid gland. You can see in the figure how calcium moves between bone and blood all the time. Whenever the level of calcium is down or low in the blood, parathyroid hormone becomes active. It activates or stimulates the osteoclasts, and the osteoclasts would cause bone resorption. That means it breaks down the bone matrix and releases calcium into blood. On the other hand, when the level of calcium is higher in blood, calcitonin hormone is secreted by the thyroid gland. Calcitonin inhibits or it stops the working of osteoclasts. So osteoblasts become active and they create more bony matrix using the extra calcium.